Okay, so this will be our last in-person class, unfortunately. But lots of you are from the beginning to right now. So I hope you enjoy the class, and we should teach you more, but unfortunately we can't do it. But something will move online. And I have already uploaded a video which is to talk about immunology. I just made it in the morning. And we're going to have another two lectures video talk about is virus and immunology. We'll talk a little bit about the COVID-19 there, so you can go ahead to watch, which is related to that, okay? A little bit, but basically talk about virus and immunology. Now, let's talk about today's lab. Let's go over some of the results. First thing is the demo. The demo, what I did is antibiotic resistant. So let's say what we had is streptomycin. And I said we have a different standards. This is 10 microgram. And we write R is less than 14. We have I is 15 to 20. And we have S is more than 21. This is all millimeter. Okay, so let's give you an example. I just gave an example. What I have is, this is the one what I made Tuesday or Thursday. I forgot about it. And you can see it's very clear. There's an inhibitory zone there. Okay, you can see it. And I also want people to see in the video. Then we're going to use a ruler to measure. So we just use a ruler to measure the diameter. Let's give an example, streptomycin. So we measure it. We're going to find it's S10, so we write here, we measure it. If we measure the diameter, you can see it is 22 milligram, 2.2 centimeter, so 22 milligram. And usually we also measure it again once we turn around 90 degrees. We do twice. This is about 23 milligram. So what we get here is we got, this is E. coli. So we get this E. coli inhibitory zone is 22 to 23 millimeter. So I will say this is fits this category. Is that right? So we say this E. coli is susceptible to streptomycin. Streptomycin 10 microgram. Okay, now does not mean that streptomycin 10 microgram you can automatically to use for the patient. You still have to use, which is twofold serial dilution to determine the eventual MIC. Because possibly it might be only 8 microgram will be working. Well, we should use 8 microgram. You should not use that one. One side, it could be cause antibiotic resistance if we use too much. On the other side, it may have a side effect for the patient. The very obvious side effects for the patient is they have the fever, because the normal bacteria could be killed using high dose of antibiotics. And the normal bacterial flora in the intestinal has been disrupted, then could cause problem. Okay, so be careful. So that's why I say it's a semi-quantitative test. Not really say that number we could be using. We only gave you an idea. E. coli is susceptible to 10 microgram streptomycin. Okay, so that's what we're going to do a demo for you. And you can see it's more obvious. Let's see, we have here a tetracycline. You see the tetracycline number. I'm not going to go very detailed. I can measure it. This tetracycline's number is probably about very small, about like 12 maybe, 12 millimeter. So when you compare here, you obviously can see it's less than 40 millimeter. So looks like the tetracycline, the E. coli is resistant. Okay, so you can see I just grab the E. coli from the lab and I put tetracycline this because there we can't find one resistant. So that means it's widely existing in the environment and everywhere regarding this antibiotic resistant. So main reason is being using too much since 1950. Okay, people using everywhere, they finally cause those resistant problems. So just to let you know that. Okay, this is the first thing I want to mention. The second thing what I want to mention is we did the sample of the food. So food sample for, we say the total microbial population. Now, it's very important, we want to do a calculation. Okay, so the steps how we do it is the first thing. The very normal, you have 10 to the minus 1, 10 to the minus 3, 10 to the minus 5. I better say you not find anything grow here, not anything grow here. You may find something grow there. Is that right? Very obvious, you should have something grow there. Now, anyway, at this moment, you have to count. Then you count it. And the acceptable zone is 30 to 300 CFU. But here, let's just give an example. Let's say I had 100 CFU on agar plate, which is 10 to the 1 dilution. Now I want to determine how many bacteria is on hot dog and how many bacteria is in ground beef. Because these two are a little bit different, if you still remember. So let's do a calculation. Now for the hot dog, you need to remember, we did that shake, and we take it out this large on the surface. So the first thing what we have to do is the measuring the surface, the area. But we don't have it. You can use a ruler to measure. I just want to tell you, approximately, it is about 22 square centimeter. Okay, now the ground beef, of course, when we did it, is about approximately 25 grams. Now, all of them is immersed in 100 ml nutrient broth. This is also 100 ml nutrient broth. So the calculation is actually a little bit different. So let's do the hot dog first. How we do a hot dog? You had 100 CFU multiplied by 10 to the first. That is how many bacteria? CFU per ml, the concentration. So that's CFU per ml. Okay, how many we get from the solution? What is the total volume of the solution? Is multiplied by 100 ml. Because that's what we get. So this is the total bacterial population what we have in the bag. Then where it comes from? It comes from the hot dog surface. What is the area of the surface? The area size, 20 square centimeter. So how we do it? This is the typical mass. Okay, approximately. You take this out, approximately 4.6. So we get 4.6 multiplied by 10 to the 3. Okay, this is 10 to the 3 CFU per square centimeter. Now I said we're going to change it to the log, so we transfer to the log. How we do it? Do a log 10, 46,000 CFU per square centimeter. What you get? This is if you do a log, approximately might be 3 to the 6 log 10 CFU per square centimeter. And I tell you one thing, that's very normal. Usually on the bacterial surface, there is about 3 to 4 log cells on the hot dog surface. So that's very normal if we get that number. Okay, now how about the ground beef? Do you want to make sure that the video camera is good? Now, how about the ground beef? This is a little bit different because we homogenize it. Is that right? So we give an example. We still see 100 square CFU. Then how we do it? The calculation will be, I use another color, 100 multiplied by the 10 the first. Okay, that's CFU per ml. Okay, now since we homogenize, so we think this ground beef and this volume is combined together. Therefore, here you're gonna be 100 ml and add 20 grams. Now, if you learn physics, you know this ml equals gram at this moment. So we can add them together. Then where are those come from? Those will come from this 25 gram ground beef. So we divide it. Then what we get? You take out these, this equals 5, so we get 5 multiplied by 10 to the 3 equals 5,000 CFU per gram. And then we again transfer them to the log 10, 5,000 CFU per gram. Then we get approximately 3.7 log 10 CFU per gram. And usually when you get a ground beef from a market, it's about 3 log. So you don't expect the ground beef, there is no bacteria there. There's lots of bacteria there, about like 3 log. Now, at one level, we will say that ground beef is not really good if it's more than 5 log. Then that ground beef is not good. Start to have a sporage. Okay, that's why this type of the calculations give you an indicator. We call it an indication of microorganism. So that's the reason. Okay, this is what we talked about. We finished talking about last week. Okay, today's work will be very easy, but it's important. We will do bacteria analysis we get it from the pound of water. So we get a pound of water. Do a water sample. Okay, so we want to discuss several things. First of all, in the United States, if you turn on any of these tap water, ideally you can drink it, because EPA had a regulation is 0 tolerance for E. coli and the coliform of drinking water in the US. Now, of course, you're going to say about chlorine, the soft water, those are chemical issues, not microbiology issue. From the microbiology standpoint, it's 0 tolerance for E. coli and the coliform. So ideally, you can drink it. But not really, because the E. coli and the coliform most of the time will be indicate a fecal contamination. And that could be coming from a human being, most of it come from the animals. Okay? Now, a long time ago, they used coliform to indicate fecal contamination. Now, right now, they more prefer to use E. coli. And here we talk about is generic E. coli. It's not E. coli 057H7. So, E. coli and the coliform, we usually call it an indicator bacteria. Okay, now, since we said drinking water is good, can be drink potable, then they, of course, they have to go through the testing. Now they go through the testing. They usually do 2 things: testing E. coli coliforms, and then they also testing another thing called fecal coliform. Now, fecal coliform basically is very similar to the coliform. They both of them coming from intestinal, and it's gram-negative, large, non-endospore forming, facultative, lactose fermentation positive. That's all the same, but what's the difference between that? It's a storage temperature, incubation. The fecal coliform we incubated at 44.5 degrees Celsius. A common coliform, we usually incubate for 37 degrees Celsius. So the temperature difference is a major difference between fecal coliform and the coliform. Okay? Now the third thing I also want to mention is that sometimes we also using to testing total microbial population, like we did in the food sample. So they're called total bacterial population. Now what we really do, we not use those plates we do. We are really talking about these aerobic plate counts. Okay, they're called APCs. This is also user indication of the how many population in the water. Okay, so these are something I want to mention. Now, what are we going to learn today? Today we will learn is a method called 3M. It's a company, 3M Petrified Analysis. So, we talk about these agar the whole semester. But agar has to be prepared. There is something will make it very convenient for you. That's called 3M Petrified. What it looks like. A paper media. It's just a piece of paper. And they have a cover. And usually what they do is all the media is dehydrated in this paper, and you add a solution, you cover it, you do the incubation, they will show up. So I think it's a very magic one. And this is for APCs, the aerobic plate counts. Now how about the coliform and the E. coli? We have another one. This is more good. There's a well inside which is containing medium, and you add a solution there, then you cover it, you solve the problem, then you do the incubation. So that's a very easy and a very nice test. That's why I want you guys to do a practice. And it's widely used in the food industry and the water industry to test the coliform and the total microbial population. Okay? So here what we talk about, this is a dehydrated paper medium. So I always say it's a dehydrated paper medium. Okay, now I want to mention what's the major difference between these and agar plates. All these paper media, we will be adding 1 ml solution versus 0.1 ml onto the agar. This is a major difference you should know. Well, this is the last lab. We want to check whether you did not good. Lots of the time. Let's say we meet on Tuesday again. Some of you will get a tiny corner in the plate. Some of you get a large size of the liquid. When I see it, I will say that person did not good because you only add 0.1 ml onto the paper media. But you shouldn't do that. You should add 1 ml solution here, because that area can fit 1 ml solution. Now, why this is important? This means the final dilution of Petrified equals final dilution of cubes. Remember, we did the cube, we added 0.1 ml there. I said that will be 10 times more than the tube. But here they should be the same. Okay? Now the next question will be, how do you know it is a coliform? How do you know it is an E. coli? And how do you know it is an APC? I'm going to show you. Okay? APCs is very simple. On this picture film, you're going to see pink polymer. Okay, what is E. coli? How do you know E. coli? Okay, we get lucky, we have some of the color like this. Okay, so I should use these. APCs are pink. Now, how about the E. coli? You will find the E. coli is a little bit purple to blue color and packaging with gas. That is E. coli. Now, what is coliform? If you see some of the pink colony, that is coliform. Now, what is total coliform? You're going to add them together. That equals total coliform. Okay, so that's what is most important there. So here are the things we're going to mention. Then you're going to go ahead to do. Now, before you do, I want to let you know how we're going to do it. Do you mind to move that a little bit? Make sure it's covered. We get this pound water. Well, let's wear gloves. So I'm going to just show you one example. So we did get this pound water. Anytime when you do this water, the first thing what you should do is shake. Make sure it's mixed very well. Lots of there at the bottom. Okay? Now, of course, we're going to do a dilution. Let's give an example of 2 dilution cubes. And I want a 1 ml. Can I have a 1 ml pipettes? And also the pipettes, the box in the middle. Yeah. Now, yes, thank you. Now, of course, we're going to do a dilution. Today we do the dilution for APC. We will do 10 to the 3 and 10 to the 5. I'd better tell you there's a lot of bacteria there, although you're not going to see it, okay? But how about the coliform? We assuming they may have some fecal contamination, may not, so we do 10 to the 1 and 10 to the 3. So here, let's do it. For coliform, E. coli, let's do 10 to the 1, 10 to the 3 dilution. For APCs, let's do 10 to the 3, 10 to the 5 dilution. Okay, now how are we going to do it? You figured out the dilution, you already know how to do it, you do it. Let's say, oh, that might not fit. These type of tips might not be fit. Yeah. So can I have the smaller one, the one in the middle? Now, the first thing what you do, when you do the dilution, you still use 100 microliter, but when you add it there, you need to make sure this is at 1000. That is very important, okay? So let's see, change the tips. Let's use that tips to see whether it works. Seems like this doesn't work. No, use the blue box. Yeah, use the blue box. I think that might be work. Okay, let's see. Yeah, that works. So, Karen, can you come here to just take a picture? Yeah. So let me show you how to do it. If we use a Petrified, what you do is after cereal dilution, let's say, you're going to add 1 ml. So you can take that one, using 1 ml. And what we're going to do, just take a picture right here. We will be gently add into the circle. And then, you take a picture again, we gently cover it. Okay? Then it will be automatically moved across the circle and averagely spread. Looks like this. Okay. Now make sure there is no bubble there, because we wanted to see the bubble is generated by the E. coli. If you create a bubble there, it's very confusing. So there's no bubble there. But how about this guy? This is going to be a little bit different. Okay. So we're going to use the pipettes again. This, after serial dilution, this is the same thing. So you can take a picture right here, and we're going to be right at in the middle. But then I want you to do one more thing, very important. This is a pressure. Okay, this is a pressure. This is make sure right, and this is on the top. It's a flat side on the top. Then you have to do a cover, a very gentle cover like that. You pressed it, let it stay a little bit, 10 seconds, you take it out. It will be a very nice circle in the middle, like this. Okay? Then we do an incubation. We can put them together. But you can't stack too much. Usually, the stack is no more than 25 pieces. Okay, when you do the stack, no more than 25 pieces. So today's work is very easy. Everybody will go ahead to get 2 petrifilm of APC of coliform and 2 petrifilm of the APCs. Do a serial dilution, get a pound of water, and do the job. It's very easy. You can finish 10 minutes. And get a practice. Still give it to TA. And we'll see whether we can get some pictures. Okay, we'll see what your results looks like. And we can take a picture next Tuesday or sometime. And this will be the last lab. So the exam will be online, will be available to do sometime, maybe next week or maybe just after Thanksgiving. I will see. Now, I want you all, if you come here, your personal item in the other lab, just grab it. To check in your box, make sure the box is clean and there's no tubes, no plates, and no any personal items, because we may have another 100 students coming in the spring for the class. Okay, so that's what you do. Now, if you want to show me your lab notebook, I have the sheet here, so you can show me. I'll just click it. You can have your lab notebook, because that may be used for the final exam. Okay, because you all use open book, you need a lab notebook. So that's what we're going to have today. Okay. So this is a pound of water. And get your tube. I think how many tubes you need, maybe use 3 tubes.